malaria, dengue, yellow fever, Zika, but also Black Death, typhus, sleeping sickness.
Cause hundreds of millions of infections and hundreds of thousands of deaths.
These problems are actually increasing because of climate change.
Insects right now, they cause damages across public health, agriculture, and biodiversity.
These problems are actually getting worse, and we are losing the fight against insects.
Vector-borne diseases shouldn't be something that we still face in the 21st century.
We need new tools to control this problem in a more sustainable way.
At Biocentis, we are developing environmentally friendly biocontrol to target the most harmful insects on Earth without affecting all the others.
And we now have tools that can solve the problem.
Europe can build. What's missing is her story. For decades we got taught to fear the future, not build it. That's why we are making documentaries about the engineers, scientists and founders building the future. Here in Europe.
Still today, insects cause humongous problems across a variety of sectors. Mosquitoes are the animal that kill most people on Earth, almost 1 million people every year.
Agriculture, insects destroy 25% of the global agricultural production. And it is a problem that is getting worse over time because globalization and climate change is increasing the range of many harmful insect species.
And when an invasive species arrives in an ecosystem, it can displace native species and create a lot of problems with biodiversity.
So these are problems that, especially in Western countries, we're not too used to because they were mainly confined in the tropical part of the world. But they are becoming global problems. So now Europe has to deal with dengue fever, the US have to deal with dengue fever. Things that we thought were a relic of the past are becoming something that we'll need to face in the future.
At Biocentis, we are building the next generation of insect control products to better solve the problem in a way that is fully biological, non-toxic, more effective and more sustainable. Biocentis leverages on 2 decades of research at Imperial College London.
Imperial College is one of the global centers of excellence for genetic control.
I worked in this really fantastic team where they had this really amazing vision.
Focused on solving a very concrete, very big problem, malaria.
Malaria is a disease that's spread by mosquitoes, causes the death of more than 500,000 people each year, mostly children under the age of 5 and mostly in the poorest regions of the world in sub-Saharan Africa.
They started a very ambitious project that was aimed at developing the ultimate tool to eradicate malaria from Africa.
We're working on a technology called Gene Drive. And that technology is where you can engineer a mosquito and you can release that mosquito into the wild and the mosquito works for you. So that mosquito goes into the wild and it can spread that modification to the entirety of all the insects that can spread malaria, but none of the insects that cannot. And it means that you could control the species on a massive scale, not just in the area you release, potentially an entire country, potentially the entire continent, to protect people from malaria.
The idea behind Biocentis was let's further develop this technology, let's extend this technology for use in other fields. So what we're trying to do is massive. We're trying to solve problems that no one globally has managed to solve in a way that is fundamentally new. So this is the entrance of the insectary.
At the moment, we are focusing on 2 products. One is to control the mosquito that transmits dengue, Aedes aegypti, and the other one is to control Drosophila suzuki.
So Aedes aegypti is a mosquito that spreads the vast majority of arboviruses, and that includes dengue fever, Zika, yellow fever. We focused on this because there isn't a solution that's good enough today. And we thought that the technologies we're developing could make a real difference and save lives. The second species we decided to focus on is Drosophila suzuki. And this is very different. It's a fly. But that fly started off in Asia and it migrated, it's an invasive species, and moved its way into Europe and into the Americas, where it's been destroying berry production. So it's a really strange insect. It's able to lay its eggs inside the berries when the berries are very young. And because of that, it causes them to be destroyed too early in the season, wipes out entire crops, and has decimated the industry. And we thought we could target this one particular insect species and make a real difference to agriculture. So we aim to release the insect that is damaging to control the insect that is damaging.
engineering and releasing insect males that carry genetic traits that allow them to mate with the wild females and reduce their fertility.
To understand how that works, you need to understand a little bit about the insect itself and why they're damaging. These are some genome-engineered mosquitoes, which is quite nice. If I roll up my sleeve and I put my arm by the side, you'll see that the females start to gather towards my arm, and they're now probing. And what they're attracted to is they're attracted to the heat that's coming off my hand, they're attracted to the carbon dioxide, and they're attracted to the smells. The males are not so attracted. The males continue to swarm in the middle, they continue to fly, but it's the females that come towards you trying to bite. So, generally speaking, it's only the female insects that are able to cause the problem.
So in the case of mosquitoes, it's female mosquitoes that bite humans.
For a fly, the females lay their eggs in the berries, the males don't, and there are lots of other awful insects where the females are almost always the problem. That means that the males are not the problem. So you can release males into the environment, but what do they do? Why do you want to release males?
The idea is you want to surround females in the wild with sterile males that you have released. So you're going to release hundreds of times more males than exist in the wild to make sure that every wild female will only find sterile males to mate with.
Female mosquitoes tend to mate only once in their life. So you release the males are able to mate with the females think that they're mated, they don't mate again, but they don't produce any progeny and the population goes down.
So you can release as many male mosquitoes as you want without being worried that the insects that you release will cause a damage. Genetic engineering is precise. We can obtain the desired effect on fertility without damaging the fitness of the individual, so that the ones that we release are as able to compete, fly, and mate with wild females as the insects that are already there.
A birth control pill for men, which is something that everybody wants. In this case, we've done it for insects. The way that we do that is we use precision genome engineering, and we can use them to edit the genes that mosquitoes need to reproduce.
We take eggs, we align them, then we inject them with the construct using a microinjector.
I am currently performing egg microinjection from Drosophila Suzuki, and I'm using micro needles that are made from quartz, and the tip is under 1 micrometer.
The microinjection process is quite complicated because most of the eggs do not survive the injection. And of those eggs, even less will have incorporated the plasmid.
We have to spend time hatching those eggs and looking at them one by one to see, are they modified? How do we know which one is genetically modified? So we add a fluorescent protein, and that fluorescent protein will make those insects glow bright red, bright blue, or bright green, depending on which modification we've made. And then we can see very easily, you can look at these insects under a microscope, you shine a laser, and you can say the one that's glowing red, that's the genetically modified one. Once you've made one, then it's easy. Then you can mass produce them just by breeding them like any other insect. So these are climate controlled chambers. We keep them nice and hot. We keep them at 27, 28 degrees. It goes up a little bit, down a little bit, and 80% humidity, because that's what mosquitoes like.
So we start from the aquatic stage, which are the eggs one. So basically we put this cone in, which is a bit wet. The mosquitoes lay the eggs on it. And then when the times come, we put the eggs in these trays and they become larvae. After the stage of larvae, we have the pupal stage, and then they become adults, and we put them in cages to feed them basically with the sugar that we have here, to feed the female with the blood to have another cycle with the eggs. So we are now making the hemotech feeding with the blood. We are putting the blood in this plaque that will heat the blood to 37 degrees to simulate the human blood for them to recognize it and basically bite this plaque and take the blood meal.
The idea of using insects to control insects is not new.
So there's a technology that has been around since the 1950s. It's called the sterile insect technique. And the idea was you can take insects, you can sterilize them using irradiation, so typically X-rays or gamma rays, and you can release males in the environment.
And it had a massive success story in controlling this one particular insect called the New World screwworm, which is awful. The New World screwworm is one of the worst things you can imagine. It is a flesh-eating fly. It lays its eggs inside the skin of animals like cattle, like cows, and then it hatches inside them, eats the cattle, and causes them to die. Now, this was so bad that the US government spent millions upon millions and decades releasing these sterile males, and they managed to completely eliminate the screwworm from the whole of North America and pushed it all the way down to Panama. Now, unfortunately, those insects have broken free of that particular barrier zone, and they're now moving up north, and they're already in Mexico, and the US is very worried that this insect is going to come back.
So the reason why its use have historically been limited is that it is very expensive and it's operationally complex. So you need to rear and release a huge number of insects continuously.
The problem is they're very difficult to scale, and they're very difficult to produce enough of them for all the insects that we need to control. Some of them, it makes sense, like the New World screwworm, potentially a few other cases. For the others, you need something that's cheaper, something that's easier to mass produce, something where you can have a switch to make sure you're only releasing the males that don't cause harm, and you're not also releasing females that might make the problem worse.
So it is the kind of technology that required a specialized application by entities that are experienced in doing that. So it was a technology only suitable for area-wide approaches, mainly sponsored by governments, implemented by specialized institutions to solve big problems on insects that were particularly amenable to these kind of approaches.
So the reason why we need less insects is because our insects are fitter. They haven't gone through the process of radiation, so they can perform, live, and mate better in the wild. And also their effect lasts for multiple generations. Instead of being sterile, they have genetic traits that allow the reproduction to be disrupted for a longer period.
So over time you have an augmentation effect. Every one insect that you release will generate hundreds of modified males in the next generation.
In the past, if you wanted to do these types of approaches, you had to not only mass produce the insects, but you needed to separate the males from the females. Usually, you have to wait until they're adults to do this, which means you have to keep the insects in your lab, separate them as adults, and then ship those adult insects all over, potentially a region, the world, wherever they need to go. This is not so easy.
We can just do that using a genetic approach. So we rear the insects with an antibiotic in their diet. The moment you remove the antibiotic, all the female eggs become unviable. So that means that we do not need to wait for the insects to be adults and separate the sexes.
Our technology allows you to package those insects as eggs. So these here, this is a cluster, perhaps of 1,000 mosquito eggs. You can imagine how many of these we could package in one small space and ship them out for control. You can dry those eggs. You can have millions of them, hundreds of millions of these dried eggs, and ship them from one production facility all over the world, release them in a particular box where you just add water, and what comes out of it are these males that control the damaging population.
And this makes the supply chain much easier. In this way, we can achieve suppression in a very sustainable way without spraying any toxic substances, just relying on the power of nature.
Our technology is extremely targeted, so we develop a modified version of a single insect species, as the insects that we release will only interact, will only reproduce with those of the same species. So you perform insect control avoiding off-target effects. You will not disrupt other insect populations that you don't want to control, which is very different from what you're doing with chemical insecticides. So chemical insecticides are an old technology. They've been historically quite effective. They're relatively cheap, but they face a number of limitations. So one is they're not environmentally friendly. So they're typically broad-spectrum products that impact on the target species, but also are toxic to pollinators and the environment at large.
25,000 different insecticides that are used. You don't just kill the insects that you're trying to control, you kill all the others in the same environment. It means we kill bees, we kill other pollinators, and we kill the beneficial insects.
The other problem that they have is resistances. So over time, the few insects that are unaffected by the chemical product end up being the ones that are most successful. And what you see over time is a reduction in the effectiveness of the chemical product. So you need to continuously change the products that you use, and it's difficult to always come up with new stuff.
The ones that adapt are the ones we're trying to control. The mosquitoes adapt, the screwworm adapts, the dangerous fly adapts. What is unable to adapt as fast are the bees and the pollinators.
Our technology is completely species specific, so it will only affect the target species that is creating a problem. We can protect biodiversity because we do not affect ecosystems the way insecticides do.
I think one of the biggest reasons why we've had such great success in the last few years is that we have a fantastic team. We are a company that was born in the UK and we've been grown in Italy. I think it's important that we have some of the world's best scientists here in Europe, particularly in the UK, particularly in Italy. We have a whole bunch, and our team is a very international team. We have a really supportive team, it's a young team, and it's a team that people have sometimes come from the field, but actually we've had people who've come from completely different things. We have one member who started off working with fish. We have other people who've never seen an insect in a laboratory in their life before. And we've had people who've come from the business world who had no idea about biotechnology. And I think bringing together these very different people from different backgrounds, we've been able to do something really exciting, really innovative, and able to solve problems that on my own I would have thought were impossible.
If you look at Europe as a whole, the amount of innovation that our universities manage to generate is substantial. What we struggle with is implementation. So first of all, especially in universities, entrepreneurship is not always looked as an attractive career path. It's very difficult to convince researchers that instead of staying within academia, they should go into entrepreneurship. Universities make it very difficult to create the university spin-outs, for example. It's not a smooth process. People within Europe don't always realize what it is possible.
But you need to trust that the people developing the technologies, the scientists, the innovators, that they will cross that bridge when they get there. But they need to be supported to get to those first phases.
In your life, you're not going to have a lot of great ideas. If you're lucky, you will have one, maybe two. So whenever you have that idea, it is worth pursuing because what can come out of it can really be world-changing.
It's a time where there's lots of things moving, lots of action. There are people being very successful, there are failures, but that's fine. I think it's okay to try something ambitious, it's okay to fail, and it's okay to learn from that and to do something new.
What I would say is, if at some point of your life you find that you have a very strong passion or an amazing idea, you should pursue it.
Go do it. So where do we go next? So we built some incredible technologies, but what we really need to do is to get these from the lab into the real world. We want to expand our technology to a large number of insects that are the real problems in the world.
Our vision is a healthier and more sustainable world where communities have the tools to protect themselves from insect-borne disease, and where farmers have the tools to protect their crops from agricultural pests.
More lives saved, more food production, and a healthier ecosystem. That's our vision for the next 10 years.
For centuries, humanity has been helpless against vector-borne diseases, and now, thanks to technology, we can finally solve these problems. So it can unlock a better future for everyone.